Good morning, one and all. Myself, Dr. KATHIRESAN, Associate Professor, Department of Pharmacy, ANNAMALAI University. In this video session, I am going to discuss about pharmaceutical validation. The overview of today presentation is introduction to validation, types of validation, elements of validation, cleaning validation, and analytical method validation by using HPLC. Let us say before start the session, this validation is coming under the Department of Quality Assurance Department. Let us say Quality Assurance Department have wide interlink with all the departments of the pharmaceutical industry. One is Purchase Department, Quality Control, Production, Engineering, Validation, R&D, Regulatory Affairs, and Administration and HR. So, Quality Assurance Department have wide link with all the departments of the pharmaceutical industry. Let us say first, why to validate? Because the pharmaceutical industry using expensive materials and sophisticated facilities and equipments and highly qualified person. The efficient use of these resources are very necessary, because if any failure or rejects or reworks or recalls or complaints, it will affect the production cost significantly. So, we need to validate each and every step for maintaining the quality and safety and efficacy of the product. I want to explain simply what is validation. Validation is nothing but, if you split the word VALIDATE, is very arduous, lengthy, involved, detailed attempt to test everything. It means that arduous is very difficult to achieve. It is a lengthy process. You cannot tell that I can start the work at 8 o'clock, I will finish it at 4 p.m. It is not possible. It is a lengthy process, and you need lot of involvement, because when you start with a HPLC, if you want to do any analytical method validation in HPLC, it takes around 60 to 75 injections. So, one injection it will take around 10 to 15 minutes run time. So, you need to put your energy for 8 to 9 hours with full involvement. The next one is called as detailed. Detailed of the report is very important for getting the approval from FDA, MCC, TGA. So, attempt to test everything, each and every point you have to test, and you have to test everything, and you have to submit your report. So, simply I am just explaining what is validation. Here I am just giving the definition of validation as per US FDA. Why I am giving very particular about US FDA definition means, because US FDA is a parent for validation. The rest of things are the derivatives of US FDA. So, I am just giving the definition of US FDA validation. What is the definition is establishing document evidence which provides a high degree of assurance to a specific process will consistently produce the product meeting. It is a predetermined specification and quality attributes. What is the meaning of this definition is establishing. Establishing is nothing but creating the document. What document means? Suppose you are coming to the laboratory, we are just giving the manual. From the manual you can find it out what are the experiments for this semester. From the experiment, you can find it out what is the aim of the experiment, what is the objective of the experiment, what is the procedure, how to write the report, all the things will be there. This is establishing, means we are just creating the document evidence which provides, means after giving your after giving the manual to you, after reading that you know what are the experiments for this semester. So, what is the name of the experiment also you know that. So, from the experiment you are just doing the experiment and you creating the evidence. What is that? Is observation. From the observation we are just showing that what is given in the manual, you did the same protocol and you are getting the product for that. So, establishing is nothing but creating the document evidence which provides a high degree of assurance to the specific process. It will consistently produce the product meeting. So, what is the protocol given to you? If you follow the same thing, it will meets the product. If you not follow the protocol, it will not meet the product. So, it is a predetermined specification. We are not doing anything, it is already existing. Means existing methods means it is already given in the pharmacopoeia, USP, IP, extra pharmacopoeia, Martindale. The protocols are given in the particular pharmacopoeias. We maintaining the same quality what is prescribed in the pharmacopoeia. So, this is the definition of FDA. Next one is, what are the approaches normally the practicing in the pharmaceutical industry, especially in validation. First one is called as prospective validation, second one is called as retrospective validation, third one is called as concurrent validation, and fourth one is called as revalidation. What is prospective validation? Prospective validation is nothing but pre-planned protocol. when it is required. Whenever you launching anything new to your organization, for example, equipment or facility or process, that should be validated before going to the routine pharmaceutical formulation commences. It means that if you create any facility in your organization, that should be validated before going to the routine. That type of validation is called as prospective validation. What is retrospective validation? Retrospective validation is nothing but review and analysis of historical data. Normally, when the PG student goes to the industry, especially in quality assurance department, they used to give this type of validation is called retrospective validation. What is retrospective validation? Suppose in 2020 to 21, whatever the process happened, they will give the file to you, and based on the file, you have to take the data. From the data, you have to show that the data is converted into graphical presentation. From the graphical presentation, you can show that your process always within your control. So, this is called a retrospective validation. Retrospective validation is nothing but review and analysis of historical data. Through that data, through graphical presentation, you just showing your process always within your control. Next one is called as concurrent validation. What is concurrent validation? Is continuous monitoring of the critical step involved in the process. For example, if you take tablet manufacturing, what is the critical step? Is granulation. Why it is critical step? If you not meet the granulation properly, your practical yield will be question mark. So, suppose I am giving for 100 tablets, I am giving the raw material for 100 tablets. If your granulation is not perfect, you will reach only 80 tablets. So, there is a question of 20 tablets question mark. It is 20 tablets means directly linked to the financial. So, concurrent validation is nothing but continuous monitoring of the critical steps. So, if you take tablets, granulation is the critical step. If you take the capsule, the polishing is the critical step. If you take liquid orals, mixing is the critical step. If you go to sterile product, maintaining the sterile condition is the issue. If you go for aerosols, the maintaining the dose content in the last spray is a critical step. So, what they will do, in the critical step, in any problem arises, you should resolve in the particular step. That type of validation is called as concurrent validation. So, it is simply explaining the critical step involved in the process. Next one is called as revalidation. Prospective validation is whenever you launching anything new that to be validated, that validation is called as prospective validation. When you go for revalidation, if you changing anything, that to be validated, that validation is called as revalidation. For example, laminae airflow chamber is installed means newly installed, you go for prospective validation. If that fan is not worked, I replaced the fan, that is go for revalidation. Next one is called as various types of validation. What is the various types of validation? Is one is equipment or instrument validation. In academically, we called as instrument validation or equipment validation. When you go for pharmaceutical industry, they use the terminology is called as qualification certificate. Normally, DQ, IQ, OQ, PQ, these 4 certificates are required to start your process. DQ is a design qualification. The design qualification is nothing but whatever is required, you just giving to the engineering and you just fulfilling your requirement, that is called as design qualification. For example, if a reactor, ready-made reactors are available for only 200 liters or 300 liters, but you need 250 liters means you just asking your requirement to engineer for designing that. That type of qualification is called as a design qualification. Rest of things, the pharmacy fellows will deal with the IQ, OQ, PQ. One that is called as installation qualification, operational qualification and performance qualification. And next one is called as area qualification, and next one is analytical method validation, cleaning validation and process validation. Now, I am going to explain what is IQ. What is installation qualification is nothing but suppose you are purchasing any new equipment. For example, if you are purchasing HPLC, after getting the HPLC, there will be the manual, installation manual will be there. That manual, as per manual, you have to install everything. So, how to connect the pre-injector, injector, column, everything you have to connect as per the given instruction. Suppose you are purchasing the tablet machine, 28 station machine, you have to get the manual and according to the manual instruction you have to install that. Means once you purchase, if you want to install in the first floor, whether it is capable to install in the first floor or not, you have to ensure that. So, as per installation procedure, you have to do the installation qualification. Then operational qualification. After installation, you have to operate the system and whether it is what you giving the instruction, whether it is obeying or not. For example, if you purchase the hot air oven, if you maintain 100 degrees centigrade, after giving the instruction to the 100 degrees centigrade, if you enter the 100 degrees centigrade, whether 100 degrees centigrade is maintaining or not inside, you have to check. How to check? By using the probe test. You have to keep the different probes in the different location and you have to find it out what is the temperature of the each probe, whether 100 degrees is maintained or not. The next one is called as performance qualification. What is performance qualification? After purchasing, you just giving the certificate is called as operational qualification. Operational qualification, after 1 year, whether it is performing the same or not, you have to ensure. For example, if you have in your pharmaceutical industry neocoater coating machine, it may be the 100 RPM per minute, but while purchasing it will be 100 RPM. After 1 year, due to continuous process, there may be the rotation speed may be reduced. So, you have to do the performance qualification. In performance qualification, after 1 year or 6 months, depends upon your batch size, you have to do the performance qualification and ensure that it is the same 100 RPM is maintained. If it is not maintained, only 99 is coming, means we have to change in the SOP. In standard operating procedures, we have to instruct this is only 99 RPM. For example, if it same thing for HPLC also, while purchasing it may be detect 0.1 ppm. The detector capacity can detect the 0.1 ppm, but after 1 year, whether it will detect the 0.1 ppm or not, you have to ensure that. How to ensure that? After that, you have to slowly reducing the concentration of your sample and you have to ensure whether it can able to detect the 0.1 ppm or not. If it is not able to detect it, only up to 0.5 ppm is detecting, you have to change in the SOP. This instrument from this date it is capable to detect only 0.5 ppm. Next one is called as DQ. DQ actually it is not coming under the pharmaceuticals, we just giving the what is requirement to the engineering. And next one is called as elements of validation. The validation of the process. Suppose if you want to do the process validation, let us see what are the elements are required. This is the common elements. I am not very specified to the particular process. I am just giving the instruction about generally what are the elements will be there in for doing the process validation. Next one is called as, this is suppose you want to do the process validation, these are the things you should know before start the process. First you should know about the instrument calibration, how to calibrate the instrument. Second one is called as critical support system, means what are the critical steps involved in the process you should know first. Then third one is called as raw and packaging material. What is meant by raw and packaging material? What is the raw material and what are the excipients you are using, and you have to collect all the report from the R&D department, whether raw material have any interaction with the excipients, all the details, you just ensure that. After that, you just go for product designing, whether it is a white color bottle or amber color bottle, what is your design, what is your aerosol designing, everything comes under the product design. Next one is called as manufacturing stages. What are the stages involved in the manufacturing process? Then you should know what are the equipments they are using and what are the facilities they are using. Next, you have to qualify the person, that is called as operator qualification. The operator qualification, you just give the instruction to the operator, what are the steps to be followed as per standard operating procedure. While doing the manufacturing, they should follow the batch production record. Next one is called as analytical procedures. Analytical procedure, what analytical procedure? In-house specification or as per pharmacopoeia you following, you have to collect all the information, then only you have to start the process validation. Next one is called as cleaning validation. How to do the cleaning validation? So, for reading the cleaning validation, you have to spend around 20 pages you have to read, then only you can get the conclusion. Normally, when PG students go for industry, if they get a cleaning validation for 6 months period, they do not know how to start the work or how to end the work. Because the reason, because it is an interdisciplinary work, you have to work in the quality assurance department, quality control department, production department and documentation department. So, the fellows, those who are working in the industry, newly going to the project, they do not know how to start the work. So, if you are going for project work, if they are giving the cleaning validation, so your 6 months duration to be divided into 4 phases. One is called as a development phase, another one is called as planning phase, third one is called as execution phase and the fourth one is called as a maintenance phase. If you not complete at any one phase, you cannot submit your report completely. So, in development phase, first phase, suppose if you have 6 months, you have to take for 1.5 months, you have to work in the development phase. In the development phase, first thing is analytical method development. Whether method is developed or not, you have to ensure that if method is already developed means you have to go for next step is called as analytical method validation. Analytical method validation I will give you in detail in the next slides. For analytical method validation, you have to do in two way by using USP guidelines or ICH guidelines. If it is first phase is completed, you just go to the next phase, that is called as planning phase. What is planning phase? In the planning phase, which equipments you are going to clean? In the equipment, after selecting the equipment, where is the sample site, where you are going to collect your sample. Second one is surface area. You have to calculate the surface area of the equipment. Then you have to collect the schematic diagram. First, in third step in the planning phase is, especially in the equipment process, you have to collect the schematic diagram of the equipment. Then only you can calculate the surface area, then only whatever you collected the sample, that sample result you can convert into the total surface area. Next one is called as analytical selection and acceptance criteria. So, you have to which method you are going to your validated method, after selecting that you have to fix the acceptance criteria for your samples. So, what cleaning agent you are using, what is active agent you are using. For example, if you in your production unit, if you are preparing the paracetamol, normally they are using the cleaning with cold water, but paracetamol is highly soluble in hot water. So, if you replace the cold water to the hot water, minimum you can save 100 liters to 150 liters water. So, these are the things you have to include. So, for that you need the drug profile, whether drug is highly soluble in which medium. So, depends upon that you have to select the cleaning agent. Next one is called as protocol development. After doing these two steps, you have to develop the protocol and you have to write the protocol and you have to get from the approval from your higher authority and you have to train the individuals. After that, it will become, once it is approved, it will become as a cleaning SOP. So, the cleaning SOP after writing and approval, you have to train the individual, those who are going to do the process. Next one is called as execution phase. Once the protocol is executed, you have to clean and you have to collect the sample, and after collecting the sample, you have to test everything. If it is test passes, you just go for validation report. If the test is failures, you go for incident investigation, that is called as root cause analysis. You have to find out where there is the reason for failure. If it is not failed, you just go for a validation report and you just go for continuous monitoring, that is called as maintenance step. If it is changing anything, if you want to change anything in your setup, you have to go for revalidation. So, this is about the cleaning validation. And next one is called as analytical method validation by using HPLC. I am very particular about HPLC means when you go for UV, the steps are little bit minimum, but if you go for HPLC, it is precise also. So, I am just giving how to validate the analytical method by using HPLC. Here the definition, normally in FDA telling the analytical method validation, simply it is called as 8 steps of method validation. And sometimes they call it as in some guidelines it is mentioned as analytical performance parameter. In some guidelines they mention analytical figures of merit, especially if you go for MCC, Medicinal Control Council, and TGA, Therapeutic Good Administration for Australian guidelines, they will mention as analytical figures of merit. When you go for International Conference of Harmonization, that is ICH guidelines, they will mention in the name of validation characteristics. So, this is the steps normally involved. Normally we follow analytical method validation by 2 guidelines, one is USP parameter, another one is called as ICH guidelines. What is the difference between these 2 guidelines is, one is as per USP, accuracy, precision, limit of detection, limit of quantification, specificity, linearity, range, ruggedness and robustness. What is the difference between ICH and USP is only system suitability. System suitability is the only parameter added, but in USP that is we doing in the specificity. So, the difference in the USP and ICH terminology is for the most part one. However, one notable exception, that is ICH treats the system to suitability as a part of method validation, where the USP treats in the separate chapter 621. So, discussion of definition of analytical performance parameters are given below. So, first accuracy, how to do the accuracy? It is a measure of exactness of the analytical method, means you just take the sample, you inject for 6 times, and whatever you are getting the value, whether your value is how close to your true value or your reference value. that you have to calculate, that is called as accuracy. I will tell you the practical procedure also how to do the accuracy. So, simple whatever drug recovery percentage, normally they use in industry, it is called as recovery percentage for accuracy. You have to take just 25 milligram of working reference standard drug, and you just put it, after taking the weight, you just put it in the 50 ml of volumetric flask. Before taking the volumetric flask, you just ensure the volumetric flask is clean and dried. After that, you just make up with the mobile phase up to 50 ml. So, that is considered as a stock solution. From the stock solution, you just taking the 1 ml of stock solution, you just spiking the concentration in 4 different volumetric flasks. One is labeled as 0.0, that is first one. Second one is 0.2, means from the 1 ml stock solution, you just take the 0.2 ml of standard, you just make up the volume. Like that, you just slowly spiking the concentration and injecting that and calculating the recovery percentage. After getting the chromatogram, you just calculating the recovery percentage. Second one is called as precision. What is precision means? How precise your test is, you have to check. In the same way, you have to prepare the solution and you have to inject the 6 times, whether the same reading means repeatability is there or not. You have to ensure that, and you have to ensure with the statistically also RSD and with the help of SD, you have to prove that it is precise or not, you have to ensure. I will tell you the procedure how to do that. Normally, the precision, according to the ICH, the precision should be performed in the different levels. One is repeatability, another one is called as intermediate precision, another one is called as reproducibility. In ICH, it is not mentioned in the USP, but in ICH, it is mentioned in the different 3 methods. So, what is repeatability? It is nothing but inter-assay precision. Is a result of method operating over a short time interval under the same condition. I am developed the method, I am in the short interval, I am just doing this experiment and I am getting the result. Whether I am getting the same values or not, I have to ensure. Second one is called as intermediate precision, is the result from within the lab variation due to random events such as different day, different analyst and different equipments. Means after developing the method, I am just giving the method to one more person as to do in the different version of the HPLC. They are doing the same, by using the same protocol, they are doing that. After getting the result, you just comparing the result with your instrument and another instrument. Whether the result is precise or not, you have to ensure. Third one is called as reproducibility, the result of the collaborative studies. You just, whether the results are reproducible or not, you have to ensure. For that, ICH guidelines, it is mentioned in the 3 way. So, it's procedure, you have to again take a 25 milligram of working reference standard in the 50 ml volumetric flask. From that 1 ml stock solution is prepared. From the 1 ml, that what is the concentration of that stock solution is 10 ppm. From the 10 ppm, you have to inject 6 times. From the 6 times, you have to calculate the area. From the area, you have to take the values and you have to do the RSD and SD, and you have to ensure it is reproducible. Then specificity. How you are specific in the test. Specificity is the ability of to measure the accurately and specifically the analyte of interest in presence of other compound. For example, some impurities may be mixed in your compound. So, if you want to identify your compound only and you want to show that this is my compound, this is impurity, if you want to tell, you have to perform the specificity. It is otherwise called as impurity analysis. So, here what I am just giving the, here I mentioned the drug name also. The drug name is terbutaline sulfate, and what impurity I have used is 3,5-dihydroxy W2-butyl aminoacetophenone. So, I am just mixing together both drug, my drug, as well as the same, in the same time it is the impurity also elute. So, I am mixing together and I am just injecting to the HPLC, and I am taking the 2 peaks. From the 2 peaks, I am just telling that this is my drug peak and this is the impurity peak. So, LOD, next one is called as limit of detection. What is limit of detection? Suppose I am taking from the stock solution, I prepared the 10 ppm solution. From the 10 ppm, I am slowly reducing the concentration from 10, 9, 8, 6, 5 up to 0.1. How long I can reduce means, once I sample is injected, I can able to find the peak, but I am not able to calculate the area. So, that is called as limit of detection, means I am just finding the capability of the detector, how much it can able to detect. So, what is that is I am slowly reducing the concentration from the lowest concentration. At the lowest concentration I can find out the peak, but I am not able to calculate the area. Simple example, I can by visual checking means I can see some distance one fellow is moving. So, one fellow moving I can able to assess, but I am not able to assess who is that. So, that is called as detecting capacity. Object is moving, I am identifying that. So, that is detecting capacity. Here also we just assessing the detecting capacity of the instrument. So, how to assess the detecting capacity? I am just reducing the concentration from the lowest concentration at particular point, it will show only the peak, but not area. So, that is called as limit of detection. If you want to calculate the signal to noise ratio, it usually comes the what is the standard limit for the 2 to 3, means 3:1 or 2:1. So, the ICH has recognized the signal to noise ratio convention, but also list two other options to determine LOD, visual non-instrumental methods are the means of calculating the LOD. So, you have to calculate by manually also, you can calculate by lowering the concentration and taking the chromatogram, and manually you just putting the baseline, from the baseline you just calculating the area. So, what is the procedure is again 25 milligram of working reference standard is taken, and you just preparing the 1 ml, that is working reference standard drug is taken, and you just adding the 50 ml of mobile phase. From that you just taking the 1 ml, from the 1 ml you just make up to 50 ml. So, what is the concentration if you ask, that is 0.1 ppm. So, from 0.1 ppm I am injecting 6 times, and I am finding that I am getting only peak, but I am not able to calculate the area. It means that is the LOD of the sample. So, what is LOQ? Limit of quantification is nothing but like that. In the same way you have to the next step, LOD means you can find the peak, but area you are not able to calculate. If you slowly spike the concentration of the sample from 0.1 to 0.2 ppm, in 0.2 ppm it is showing the peak as well as area, means that is the LOQ. Means if you reduce the concentration, it will not show the area, means that is LOD. If you spike the concentration, it will shows the peak as well as area, that is called as LOQ, that is called as quantification. I can able to quantify the amount of drug present in the given sample. What is the signal ratio limit is 10:1. And this is the procedure again, you have to take the working reference sample, means it is normal, this is a common stock solution. From the stock solution I am just taking the 5 ml, from the 5 ml you just make up to 100 ml, means the concentration of my sample is now 0.5 ppm. So, 0.1 or 0.2 is a LOD, 0.3 also for example 0.5 ppm I can able to get the peak as well as area, means 0.4 ppm I am not able to get the area, means the 0.4 ppm is LOD and 0.5 ppm is LOQ. And linearity range, what is linearity and range is just I am spiking the concentration slowly, 0.1 ppm, 0.2 ppm, 0.5, 0.10, whatever the values I am getting, I am just plotting the graph. After plotting the graph, if I just line up the points, if I get the linear line, that is called as linearity. What is the range is, what is the lower concentration, what is the upper concentration, that is called as range. So, range is the interval between the upper and lower level of the analyte that have been demonstrated to be determined with the precision, accuracy, linearity using the method as written. So, next one is a procedure. So, from the stock solution you just spiking the concentration 0.4, 0.6, 0.8, 1, 1.2 and 1.4. So, you just labeling that, then you just make up the volume with 50 ml by using the mobile phase, then injecting the just take the sample and inject for 6 times. From the 6 times you just taking the value, average value. After taking the average, you just plotting the graph. After plotting the graph, if you join all the points, if you get linear, that is called as linear. What is the lower and upper limit is called as range. Ruggedness, what is ruggedness? Ruggedness is how it can able to reproducible. So, RST to be calculated, I am just giving the estimate to other person in the different laboratory, different analyst and different instrument and different reagents and different day. So, I am just handovering my experiment procedure to the other persons in the different laboratory, they are performing everything. After getting the report, I am just comparing the report with my report, that is called as ruggedness. It should not be deviated. So, ICH did not address the ruggedness specifically, instead it covered the topic of ruggedness as a part of precision, because in ICH they mentioned in the precision itself, but if you go for USP guidelines, it is mentioned in the ruggedness. So, what is the procedure? It is nothing but different analysts carried out the performance of the method on different days on different instruments. So, robustness, what is the robustness? Suppose if you slightly, because mobile phase once prepared, next step while preparing there will be the slight change in the pH, that slight change in the pH will not disturb your experiment. So, next day if you preparing the mobile phase with a slight change in the pH or percentage organic or ionic strength or a temperature and determining the effect, it should not affect the result of the method, that is called as robustness. So, as in ICH, robustness should be considered early in the development of the method. In addition, if a result of method or other measures are susceptible to variation in method parameter, this method should be adequately controlled and a precautionary statement included in the method documentation. So, whatever you getting the values, that should be documented for approval. So, system suitability. What is system suitability? Is nothing but whether this system whatever you developed the method is suitable or not, you have to justify that. So, your values are able to reproducible or not, and your resolution factors are reproducible or not, you have to ensure that. So, parameters such as plate count, tailing factors, resolution, reproducibilities are determined and compared against the specifications set for the method. So, whatever this from taking the chromatograms, from the chromatograms you are taking all the data just comparing with the method, whatever you selecting the method, that is suitable for not suitable for experiment or not, you have to ensure. And this is USP chapter 1225 on validation of analytical methods, specifically addresses the terms and definition, but leaves the protocol and the methodology open for interpretation. So, this is my conclusion. A well defined and documented validation process provides the regulatory agency with evidence that system and method is suitable for the intended for the use. Means you just creating the document evidence and ensuring your quality of the product. That same document is just submitting for the approval of FDA or MCC or TGA, where you want to keep your product in which country. Based on that guidelines, you just following with the method. After from the method, you just doing the experiment, and from the experiment you just showing I follow the same method and I am maintaining the same quality what is prescribed in your norms. So, we just giving the report and getting the approval for the product marketing. So, this is my reference I used United States of Pharmacopoeia, and these are the reference used for this preparation. Thank you.